You have heard a sentence like this one, and you have probably repeated it. It worked in mice. Maybe it was a cure for something you cared about, and it was going to be 5 years away, and that was a while ago now. You did not think you were being lied to. You were not. Somebody ran a real study in a real building and got a real result, and the result was true. Then it went out into the world, and it stopped being true. That gap has a shape. It is not fraud, and it is not sloppiness. It is a place. A lab is a room somebody built, and it is one of the strangest rooms on Earth. And almost everything you know about how bodies work was learned inside one. So we are going to look at the room instead of the finding, because the thing that makes a result clean is the same thing that makes it hard to carry outside. Start with the temperature, because it is the smallest fact here, and it is the one that will undo you. There are rules about how warm a mouse room has to be, and the number sits somewhere around 20 to 26 degrees. That is a comfortable room for you because you are wearing clothes. A mouse is not wearing clothes. For a mouse, the temperature where the body spends nothing at all keeping itself warm is closer to 30. So every standard laboratory mouse in every standard mouse room is slightly cold all the time for its whole life. It is not suffering, it is spending. If you have ever sat in a room that was almost warm enough, you know the difference. In 2013, Kathleen Kokolus and her colleagues published a paper in PNAS asking what that costs. They did the simple thing, which was to move the mice somewhere warmer and watch what tumors did. They got a striking drop in how tumors formed, how fast they grew, and how far they spread. And it depended on the immune system. The mice were not worse off in the cold because cold is bad for you. They were worse off because a body that is paying to stay warm has less left to spend on everything else. Nobody wrote that in the methods for decades because it was the room, and nobody writes down the room. It gets stranger, and this is the part that should change how you read all of it. A laboratory mouse is kept clean on purpose. No infections, no parasites, no dirt, nothing. That is a good idea, and it is why the experiments work at all. But in 2016, Lalit Bira and his colleagues published a paper in Nature pointing out what a life like that actually produces. The immune system of a standard clean laboratory mouse does not look like the immune system of an adult human. It looks like the immune system of a newborn. Then they did the obvious thing, and you may find it funny that it took until 2016. They went and bought mice from a pet shop. Ordinary, grubby mice that had lived a normal life. They housed them together and the laboratory mouse grew up. Its immune system matured into something much closer to an adult. So for years, when a treatment that worked in mice failed in people, one quiet reason was that we had been testing it on the equivalent of infants who had never met anything. Now hold those two facts next to each other, because they are really the same fact twice. The cold and the cleanliness are not mistakes. They are controls. You would have chosen both of them. You keep the temperature fixed so that temperature cannot be the reason your result came out. And you keep the animal clean so that an infection cannot be the reason either. Every one of those choices makes your experiment sharper. Every one of them also moves the animal further from anything that has ever lived outside. That is the trade. And it is not a fault in the method, it is the method. Control is what buys you a clean answer. And control is what makes your answer local. You cannot spend it twice. So picture two places, and you have spent time in both of them. There is the lab, where you know everything, because you took out everything you did not want. And there is the jungle, which is only a word here for everywhere else. Nothing was taken out there, and it is all still happening at once. A finding is born in the first place, and then it has to go and live in the second one, and most of them do not survive the move. When one fails, the story you get afterwards is almost always about the finding itself. It was overhyped or rushed, or somebody wanted a headline. Sometimes that is exactly right, but very often nothing went wrong at all, and the result was perfectly good. It simply came with a room attached. Nobody carried the room out with it. We are all bad at that last part, and you can watch yourself doing it. We repeat the sentence, and we drop the conditions. A sentence travels easily, and conditions do not travel at all. So, what should you do with any of this? Not distrust, and not the lazy version where nothing is knowable, and you get to stop reading things. The work is how we found out about the cold and the cleanliness in the first place. Both of those were labs turning round and checking themselves. What changes for you is much smaller than that. When you hear a result, the useful question is not whether it is true. It is what was held still in order to get it. Something always was. And we should say the obvious thing about this film. We are standing in a room too, and you can see the walls of it. Every image you have watched was made under conditions we chose and then locked so that they would come out the same every time. The voice is set, and the palette is two inks. Nothing you have seen here has ever met any weather. So, take it the way you should take the mouse. It is not wrong, it is just very clean. And clean is a place, and you do not live there.